Good afternoon. We will begin. We're very particularly grateful to one of our speakers today, Dr. Raffit Hassan, who's been kind enough to come to bat for Dr. Paston, who unfortunately, due to a family problem, was unable to be with us today. So, just in, broadly thinking, what seem to be the major advances in cancer therapy in recent times? It seems to me they fall into sort of 2.5 categories. One is the use of genomics and genomic profiling to detect possible susceptibility genes, and also to lead to the development of targeted therapy based upon identification of specific genes, their protein structure, and so forth. And related to that is the development of antibodies, which seem to be increasingly emerging on the scene as being potentially powerful cancer chemotherapeutics. But the other major area is what I call here the taming of biologics, toxins to therapy. And sort of in thinking about it, it struck my attention that quite possibly birds and mammals are the only two species that, to my knowledge at least, don't elaborate biologic toxins. We do our toxicity more verbally and physically than we do by chemically, I guess. But throughout the rest of biology, the elaboration of toxins which kill by a variety of mechanisms which are not reversible. This is very different from, say, drug-related chemotherapy, which may affect a cell and give rise to alternative ways for drug resistance, for example. These biologic toxins are like the guillotine. There's no way to put the head back on once everything has been killed off. And when you think about it, reptiles, bacteria, fungi, plants, they live in a hostile world of battles going on all the time, and toxins are critical to their both protection and maybe in some instances to their own sort of biology other than just as protection. And in the bacterial world, the group of Clostridia, tetanus, botulinum, what's the other one? Cholera, Vibrio. I mean, these toxins are devastating. We understand something about how they affect diphtheria, but they affect the cell in a way, and this is the important thing, that it's all over with. There is no escape once a cell has been affected. So that's a very powerful way, but how do you direct it to a cancer cell as distinguished from any other cell, because any other cell can be killed by the toxins as well. So with these two big areas of, let's call it genomics and toxinology in relationship to cancer therapy, that's really the subject of today's Demystifying Medicine. Now, we're extremely fortunate in having, well, before I get to that, I would just point out two questions that come to mind, which you might think about because they relate to the subjects that will be discussed today. You might think about, what do these people all have in common that can relate to cancer? Now, you'll learn during the next hour and see how that has, how that relationship exists. Whoops, I had another one on here, which I seem to have lost. But the other one is, for example, we have patient A, B, and C, and they all have a cancer which histologically looks virtually the same. A is treated with drug 1 and gets a fantastic response. A is treated with drug 2, nothing happens. The disease progresses. A is treated with drug 3. I'm sorry, yeah, that's right. No, B is treated with the drug that the other fellow didn't have a response to, and he shows or she shows big improvement. What accounts for this extraordinary difference in cancers, which at least in old-fashioned pathology all look relatively the same under the microscope, but their biologic response is greatly different? So these two big areas that I've just touched on are really going to be the subject of this day's discussion. And we're most grateful because here at NCI are two outstanding investigators who will discuss these problems with us. So I'll introduce both of them to you at the beginning. The first is Raffit Hassan, who graduated from medical school in Kashmir in India, came to the United States, received clinical training in Buffalo, came here to the NCI for oncology training, was in Oklahoma for a while, then came back in 2002 as a tenured investigator at NCI and worked in the laboratory of which Ira Paston was the branch chief. And Dr. Hassan has been largely responsible for this toxin-related, immunotoxin-related treatment of the disease that all those people on that list that I showed you are suspect of potentially having, and that's called mesothelioma, which we'll hear much more about. And our second speaker is Paul Meltzer, who received medical training, I believe, in Kansas, and a PhD at Caltech, and is widely known for his work in molecular biology, particularly genomics. And here, he's the head of the molecular genetics section in the National Cancer Institute. And those of you who've read or will read some of the references we've put up on the website will realize that his major interest is in understanding the use of genomics to detect differences in responses of individuals with different kinds of tumors, particularly sarcomas, but many others, and to try and use genomics for the better understanding of perhaps of both, well, of diagnosis and of development of therapy. So the first speaker is going to be Dr. Hassan, and thank you very much for being with us.
Okay. Thank you so much for the invitation. So I'm sure you all were looking forward to hearing Ira, but Ira sent his apologies. I'm going to talk about our immunotoxin therapy for mesothelioma, and this is work we have been doing over the last 20 years or so. Just to get you up to speed about malignant mesothelioma, it's a tumor of the pleura or the peritoneum. And there are about 3,000 new cases diagnosed in the U.S. each year. And this disease has limited treatment options and poor prognosis. This is what we call as a PET scan. You inject radio-labeled glucose to a patient, and as you can see, As you can see, the tumor is pretty diffuse and takes up the radiolabeled glucose. So it's a pretty aggressive tumor. So mesothelioma can involve different sites in the body. It can involve the pleura, which is the lining of the lung. This is an autopsy specimen. You can see there is diffuse involvement by the tumor surrounding the lung. It can involve the abdominal cavity, where you can see these diffuse tumors all over the intestine. Occasionally, we see patients who get pericardial mesothelioma because the mesothelial cells also line the pericardium. And occasionally, we see patients with mesothelioma of the tunica vaginalis, which is the lining of the testes. So the mesothelioma can involve different parts of the body which have mesothelial cells, but the most common part is the pleural mesothelioma. In 1960, Wagner, who was a pathologist in South Africa, noticed an increased incidence of mesothelioma among asbestos miners in South Africa, and showed that this increase was because of asbestos exposure. And as you heard in the previous slide, there is increased incidence of mesothelioma in miners, family members of miners, because these miners would come home with their long coats, which were contaminated with asbestos fibers, and when they would go home, the asbestos dust would infect their family members. We also see a lot of patients who work in the defense personnel in submarines many years ago, because asbestos is a fantastic insulator and was widely used in ships. There's a long latent period between exposure to asbestos and developing mesothelioma, usually 20 to 30 years. So many of these patients usually have a remote history, but there are many patients who do not give a good history of exposure to asbestos. So there are other etiologic factors for getting mesothelioma. SV40 is very controversial, and in general the thinking is that it by itself does not lead to mesothelioma. However, there is increased incidence of mesothelioma in patients who have Hodgkin's disease or non-Hodgkin lymphoma who have got radiation in the past, and we frequently see young patients who have been cured of their Hodgkin's disease or lymphoma but present with mesothelioma. Recently, a gene called the BRCA1-associated protein has been implicated in development of mesothelioma. About 40% of sporadic mesotheliomas have mutations or deletions of this gene, but there is also a germline mutation which leads to increased susceptibility to mesothelioma. So there could be a genetic reason for developing mesothelioma, and there's a lot of interest in that. Pleural mesothelioma, which is the mesothelioma lining the lung, is the most aggressive tumor, and it has a poor prognosis. Surgery has a very limited role because most of these patients present with advanced disease, and most of the chemotherapy drugs do not work. The only drugs that work is a combination of pemetrexate, which is an antifolate, combined with a cisplatin. Even with the best therapy that we have today, the median survival of the patients is only 12 months, and almost all the patients are dead by 2 years. So there's clearly a need to develop better therapies for this patient. And our work over the last 20 years or so has focused on a protein called mesothelin. It's a tumor differentiation antigen that was identified in Ira Paston's lab many years ago. Mesothelin is a cell surface glycoprotein. The normal expression in human tissues is limited only to the mesothelial cells that line the pleura, peritoneum, and pericardium. And that's very important to develop a targeted therapy because you don't want it to be expressed in important organs. The mesothelin gene encodes a precursor protein of 71 kilodalton, which is attached to the cell membrane by a GPI linker, and it is cleaved by the protease furin to a 31 kilodalton shed fragment that we call MPF, and a membrane-bound 40 kilodalton fragment that we call mesothelin. And that is the target for our therapy. Both MPF as well as mesothelin can be detected in the serum and can be good biomarkers, but I'm not going to talk about that today. We and others have shown that mesothelin is highly expressed in many human cancers. almost 100% of epithelial mesotheliomas, as well as pancreatic adenocarcinomas, highly express mesothelin. Mesothelin is also expressed in about 70% of ovarian cancers, about 50% of lung adenocarcinomas, and other tumors including gastric, synovial sarcomas, and biliary carcinomas. So although our work so far has been focused on mesothelioma, the high expression of mesothelin in many common solid tumors makes our therapies applicable to other tumors. The biologic function of mesothelin is not known. Many years ago, Tapan Vera in Ira's lab made mutant mice which lacked the mesothelin gene, but these mice had no phenotype and they reproduced normally. We have recently shown that mesothelin is a novel CA125 binding protein and may play a role in tumor implantation in the pleural peritoneal cavity. And we just showed that MPF in mesothelin stimulates the growth of lung cancer cell line in the peritoneal cavity of mice. So this work suggests that mesothelin may have a role in tumor metastasis or tumor spread. However, our focus has been on targeting mesothelin for cancer therapy. The high cell surface expression in many solid tumors makes it a good target for antibody therapies. And expression on the normal pleura, peritoneum, and pericardium was a concern when we decided to exploit it for therapy. This is one of my first papers when I was a postdoc in Ira's lab, where we had a monoclonal antibody to mesothelin called K1. And these were athymic nude mice that had a mesothelin negative tumor in one flank and a mesothelin positive tumor in another flank. We radio-labeled the antibody to mesothelin K1 with India-111. As you can see, there was preferential uptake in mesothelin expressing tumors. And this was one of the studies that really set us on this path of exploiting mesothelin-directed therapies. So it took us about 15 years to show in patients that an antibody to mesothelin does in fact localize to tumors. This is a clinical trial that we did of an anti-mesothelin antibody called emeteximab that was radio-labeled with India-111. So this is one of my patients with mesothelioma. This is a CAT scan showing the mesothelioma. If you do a PET scan where you inject radio-labeled glucose, you can see the uptake in the tumor. And if you did a biopsy of this, you'll see diffuse mesothelin expression. And when we injected the radio-labeled antibody, it does in fact localize to the tumor. So this is a proof of principle in patients that an antibody targeting mesothelin does localize to mesothelin-expressing tumors. So we are using many different ways to target mesothelin, but today I'm going to talk mostly about our work using an immunotoxin. As you know, very few anti-tumor antibodies by themselves are good at killing cancer cells. There are some notable examples, such as trastuzumab, which is an antibody to HER2 used for treating breast cancer. Once it binds to the tumor cells, it leads to cell death. But by and large, if you take other antibodies to tumor antigen, they do not mediate cell kill. So to make anti-tumor antibodies useful, we can arm them with different substances so they become more potent. One way is you take an antibody to a tumor antigen and attach a toxin to it. That's an immunotoxin. The other way is you take an anti-tumor antibody and attach a very potent chemotherapy drug to it. These are the antibody-drug conjugates where you can deliver highly potent chemotherapeutic agents to the cancer cell. And the third approach is that you take antibodies and radio-label them with radioisotopes, which are used mostly for treating blood cancer. So a recombinant immunotoxin consists of an antibody or an antibody fragment linked to a toxin. So this is an antibody, so you take an FV and you take a toxin. The toxin that we have been working on is called Pseudomonas exotoxin A, which is made by the bacteria Pseudomonas aeruginosa. The toxin has three functional domains. The binding domain that allows the toxin to bind to the cell membrane. It has what we call as a processing domain whose function we don't know. And it has a catalytic domain shown here in the red, which ADP ribosylates elongation factor 2, leading to cell death. To make an immunotoxin, you get rid of the binding domain of the toxin and replace it with the FV of interest so that the FV directs the toxin to the cancer cell and mediates cell kill. Once an immunotoxin binds to its receptor, in this case mesothelin, it is taken up by clathrin-coated pits, goes to the endosome, the toxin is processed, goes to the ER, and ultimately to the cytosol, where it inhibits protein synthesis, ultimately leading to cell death. So SS1P is an anti-mesothelin immunotoxin. It has high affinity for mesothelin. We show that it is cytotoxic to mesothelin-expressing tumor cells of patients with mesothelioma and ovarian cancer, and it caused regression of mesothelin-positive tumors in mice. So I'm not going to go with the preclinical data, but we did a clinical trial of SS1P in patients who had advanced cancer that had failed all standard therapies. So we treated patients whose tumors expressed mesothelin. We established what was the maximum dose of the antibody that you could give to the patient, which was 45 micrograms per kilogram. And the dose-limiting toxicity, that is toxicity where we could not give any more SS1P, was pleuritis, which is inflammation of the pleura. And this is something that we expected because mesothelin is present on the normal pleura. But we just saw very minimal activity. We did not see any tumor regressions. And the main reason that we did not see any regressions was that SS1P was immunogenic. So it's a big foreign protein, and when you inject it into patients, they develop antibodies to the drug, so that if you give more drug, the antibodies clear the drug. So that's a big problem. And about 90% of the patients develop these antibodies after only one cycle. So most of the patients got one cycle and did not get any more drug. So the immunogenicity problem for immunotoxin has limited their development for solid tumors. 20 years ago, there was a lot of excitement about using immunotoxins as targeted therapy, but they failed in the clinic because of this problem. And previous efforts to overcome this problem have failed, and many felt that it's a huge problem that cannot be overcome. And that's why most of the academic centers as well as pharmaceutical industries gave up on studying immunotoxin. We have focused on addressing this problem using two different approaches. One is preventing human immune response to immunotoxin, and the second, more ambitious approach is to make an immunotoxin that is inherently less immunogenic. We and others have looked at different ways to prevent a human immune response to immunotoxins, and agents, typical immunosuppressive agents such as steroids, cyclosporin, single-agent Cytoxan, and rituximab, which targets CD20 and depletes B cells, do not work. All these patients develop antibodies. However, one of our colleagues at NCI, Dan Fowler, who works on bone marrow transplant, had shown that a regimen of pentostatin plus Cytoxan, which can deplete both T and B cells, results in host immune depletion without myelosuppression, and that it was safe in patients with renal cancer. So that was very exciting. So he gave a journal club in our lab, and we thought that we should evaluate this approach, whether this will work. So we did an experiment where we took immunocompetent mice which have a normal immune system, and immunized them with SS1P. So we gave it weekly for 9 doses. So that's a very immunogenic dose of SS1P. And all the mice shown here in the red developed anti-SS1P antibodies. So that was no surprise. But when we took a second group of mice that were treated with pentostatin and Cytoxan, none of them developed antibodies to SS1P. So that was a very amazing result, something that we had never seen before, that these two drugs could block immune response in mice. So we decided to see if the same would hold true in patients. So we decided to do a small pilot study evaluating SS1P with pentostatin and Cytoxin in patients. And the rationale was depleting the host T cells with pentostatin and the host B cells with Cytoxin would decrease the formation of anti-SS1P antibodies. And we know that pentostatin has no efficacy in solid tumors and that Cytoxin has no anti-tumor activity in patients with mesothelioma. So in this clinical trial, because this was a very different type of a trial where we're using a combination of 3 different drugs, the only patients we decided to enroll in this study would be patients who had got previous treatments, had treatment refractory disease, and had progressive disease when they were getting on the study. And our primary endpoint was to see if it was safe and feasible to use such a regimen. And the primary endpoint was to see if using these 2 drugs, we could decrease the antibodies to SS1P. So the clinical trial schema was very similar to the mouse experiment, where we take patients, give them Cytoxin by mouth for 12 days, give them pentostatin, 3 doses of pentostatin, with the goal that you knock off their T and B cells, and then you come with the SS1P on day 10 and give them 3 doses. If the patients do not develop antibodies after 1 cycle, a cycle is about 4 weeks, then they could get 2nd cycle of this regimen, but we give Cytoxin only for 4 days. So very similar to the plan in mice. So in this pilot study, we enrolled 11 patients. The median age of patients was 54. Most of them were male. 9 of the patients had pleural mesothelioma, and 2 of the patients had abdominal mesothelioma or peritoneal mesothelioma. And the median number of therapies that these patients had received before coming on the study was 3. The side effects that we saw with pentostatin and Cytoxin is grade 4 lymphopenia, that meaning that the lymphocytes decrease. And this is exactly what we wanted to do. We wanted to decrease the lymphocytes so that they did not make antibodies. And we saw some anemia and increase in liver enzyme. And the side effects that we saw with SS1P was what we expected, that they would have pleuritic chest pain because of targeting the normal pleura. So shown here is the absolute lymphocyte count of patients. So each of these is individual patients. This is the lymphocyte count before treatment, and after just 1 cycle, all of the patients had a decrease in their lymphocyte count. But at the same time, there was no change in the neutrophils. The neutrophils help us fight infection, so that was good. So the regimen was selected to the lymphocytes, but not to the neutrophils, because we were worried that these patients would be at risk for infection. And shown here are the T cells, the CD4 cells. As you can see, all the patients had a significant drop in their CD4 T cells, as well as a significant drop in their B cells looking at CD19. So both these regimens did what they were expected to do. But what was very surprising was that this regimen decreased the formation of antibodies to SS1P. As I showed you earlier in the phase 1 trial, when we gave SS1P by itself, 90% of the patients had antibodies after just 1 cycle. However, in this study, only 2 out of 10 patients developed antibodies at the end of 1 cycle. So this regimen, for the first time, could allow us to give more cycles of SS1P. But what was even better was that we saw some very good responses in patients who had a lot of disease. 3 out of the 10 patients who we could evaluate for response had a partial response. A partial response in clinical oncology means that you look at the tumors, you measure them up. If you decrease the tumor 30% or more, it's a partial response. And a complete response is that you see nothing on the CT or the PET scans. So patient 2 had a very good partial response and is still alive almost 3 years out. Patient 3, again, had a very nice partial response, as I will show you, is allowed more than 3 years. Both of these patients, as well as patient 5, had 3 prior regimen and were considering hospice care at the time we enrolled on the study, but they have had an exceptional response and alive more than 3 years. And we also had a couple of patients who had stable disease, but when they got chemotherapy to which they had not responded before, they had a partial response. Just giving an example of one of the patients, patient 3, this is a baseline CT scan at day 12, 3 months and 24 months. So this is the left lung, this is the right lung. And as you can see, you're going from the top to the bottom. You don't see any of the right lung because it's encircled by the tumor at the different place. But if you look at 3 months, basically all the tumor is gone. This is a CAT scan at 24 months. And the CT scan at 3 years looks about the same. So this was a very nice reduction in tumor bulk. Patient 5 was a patient with abdominal mesothelioma who had widely metastatic disease. As you can see, he had disease even in the subcutaneous lymph nodes. And this is after 2 cycles, the lymph nodes are gone. This is a CT scan looking at his chest. Shown here is the tumor. This is before treatment. This is his abdomen, huge burden of disease in the belly. And this is his pelvis, huge burden, disease burden. And at 1.6 months, you can see the tumor starting to shrink, and they're more or less gone at 8 months. And if you do a PET scan, as I mentioned, is you inject a radiolabeled glucose, cancer cells take it up, and they appear black. He had a lot of uptake in his neck, chest, and abdomen. And at 1.6 months, while he was getting a response, there was some worsening. But if you look at 8 months, more or less all the FDG uptake has significantly decreased. Similarly, patient 2, this is the third patient who had extensive abdominal mesothelioma. Before treatment, a lot of disease burden. There was some worsening initially, but significantly improved later on. So in summary, SS1P plus pentostatin and cytoxin in this trial was safe. It decreases the formation of antibodies to SS1P. We saw some dramatic and durable tumor responses in patients with treatment-refractory mesothelioma. And right now we're doing a small biomarker study to better understand what's going on in the tumors. So after years of working on SS1P, we're very happy that at least we are seeing some responses. And this is a patient we saw recently, again, extensive disease with a response. So this is good, but it would be better to have an immunotoxin that is inherently less immunogenic, so you do not need to use other drugs. And this has been work that is done primarily in Ira's lab to make a less immunogenic immunotoxin. So we have been using protein engineering to make such an immunotoxin. Just to bring you back to SS1P, that's the drug that we use in the clinic. It consists of an anti-mesothelin FV that recognizes mesothelin, linked to a mutated P pseudomonas toxin that consists of domain 2 and domain 3. Domain 3 is the one which kills the cell. Domain 2, we don't know what its function is. And a couple of years ago, Tom Weldon, who was a postdoc in Ira's lab, did an amazing experiment where he got rid of the entire domain 2. We thought that such a toxin would never work. But what he found is that you can remove the domain 2 of the toxin and make a toxin that we call SS1LR. And doing so, you remove most of the T cell epitopes which are present in domain 2. But the toxin still retains its activity. So getting rid of a significant portion of the toxin gets rid of some of the immunogenic epitopes, but the toxin is in fact more active. Next, Ronit Meltzer, who was also a postdoc in Ira's lab, she identified what are the B cell epitopes in the domain 3 of the toxin and mutated them to less immunogenic epitopes. And we made a molecule that we call SS1LO10, which retains its cytotoxicity but is less immunogenic. So our lead molecule consists of this portion, SS1LO10, linked to an anti-mesothelin FAB. So instead of the FV, we are using an FAB. with a less immunogenic toxin that we call P24. So all known human B-cell epitopes and major T-cell epitopes of Pseudomonas toxin have been removed or mutated. The antigenicity is reduced about 1,000-fold compared to SS1P, and we can give much higher doses than SS1P and no VLS. I'm just showing an example, looking at this new molecule, RG7787. This is cytotoxicity against primary mesothelioma cells that we get from a patient. Shown in the green is the new molecule, RG7787, compared to SS1P. It's about 3-fold more active than the parent SS1P molecule. Just to summarize a lot of the preclinical work with the RG7787, it's a less immunogenic anti-mesothelin immunotoxin. It has cytotoxicity against several primary and established cell lines from cancers that highly express mesothelin, including pancreatic cancer, lung cancer, and mesothelioma. It has single-agent activity against mesothelin-positive tumor xenografts in mice, and there's marked synergy with chemotherapy. And we have established a CRADA with Roche for this clinical development. And so this is the vial of RG7787 that has been developed, and we treated, the first patient was treated last month, and the second patient with this new molecule was treated yesterday. So we're very excited about this new molecule. And just to summarize our work, SS1P with pentostatin and Cytoxan has produced significant tumor regressions in treatment-refractory mesothelioma patients. Over new generation RG7787 immunotoxin, we believe should have more efficacy because it can be given at higher doses for more treatment cycles and have fewer side effects. So Ira and I recently summarized our work over the last 20 years about mesothelin and exploiting it as a target for immunotherapy. And I would like to acknowledge a lot of members, both in the lab and the clinic, from my group working on mesothelioma and mesothelin, and obviously Ira Paston, Christine Elewine, who's a new ACI in Ira's lab, and our collaborations with other pharmaceutical companies. Thank you.
Thank you very much, Hassan. That was wonderful. So we have plenty of time for questions, comments, and I encourage you to please ask anything you didn't understand or something you'd like to understand more.
Nice talk, Raffit. So in your new trial where you have treated with this new immunotoxin, so these patients are also treated earlier, like your first trial?
Yeah, so these are patients who have failed standard therapies.
What you see, you have some mutated amino acid. I think that just reduce the immunoactivity. But why it also makes the molecule more potent?
Yeah, so that was surprising. We really don't understand that. We thought that we might lose some activity, but in fact it is more active. I don't know whether it is with processing or how it goes into the tumor cells.
I have two questions.
Sure.
It looks like you mainly worked with the antibody fragments, right? But you were saying how they can be linked to the full antibody, and I was just wondering why you chose to work with the fragment and not the antibody.
So we mostly work with fragment, and that has, in solid tumors, we work with fragments, because if you use a whole antibody, it's about 150 kilodaltons, and you attach a protein, which is about 40. So it's a big molecule, it's a macromolecule. And there are problems with it going into the tumor itself. And so if you have a smaller molecule, such as an FV, it is about 60 kilodalton that allows better penetration. Comparing it to chemotherapy drugs like Taxol, which are probably 1 kilodalton. So these are still big molecules. So if you use the whole antibody in solid tumors, the feeling is that you may not get enough into the tumor.
Okay. And then also for your linker, does it have to be cleaved once it gets into the cell, or with toxins, does it matter if it's cleaved?
No, it gets cleaved. So the Pseudomonas toxin has a furin cleavage site. So once it goes there, it is cleaved at the furin. So when we make modification, we make sure that the furin site is there. But that's true.
Do you have any hypotheses why the patients got worse and then eventually got better from the treatment?
That's a good question. So these patients are depleted of their lymphocytes, so we don't think it may not be lymphocyte-mediated activity, but it's possible that once we give the drug, they're causing inflammation, and there may be recruitment of macrophages in the tumor microenvironment, and that's why there is increased uptake on the PET scan. So that's why we're now doing biopsies, and so we can look at what the different cell populations are there.
Why did you, wait a minute, we'll get to you. Why did you choose Pseudomonas toxin?
Oh, so that decision was made many years before I came in. So there are two major toxins there are two groups of toxin. One is the plant toxin, and the other is the bacterial toxin. So the plant toxin characteristically made from ricin, and they were very toxic in patients because of causing vascular leak syndrome. So more or less people have stayed away from them. And in terms of bacterial toxin, there are two major toxins, one made by diphtheria and one made by Pseudomonas. The Pseudomonas toxin, we know very well for the last 30 years how it works, and that's why we stuck with the Pseudomonas toxin. But they are, I think, very similar, Pseudomonas and diphtheria.
You mentioned that 50% of the patients do not have the exposure to asbestos, but they still develop mesothelioma. Do they have the same treatment after cutting by urine? Is it the same antigen or something else?
So what I meant is that about 30% of the patients have no history of exposure to asbestos, but there could be other reasons why they get mesothelioma. But that is not related to the toxin, so that's two separate things.
And a short question, after therapy with the toxin and the pentoxin, there is an increase in the tumor size. Is it because of the loss of the B cells and T cells, whatever missing they are doing?
So we think that some of it could be because there is infiltration with macrophages or other cells causing an inflammation which then settles down.
You have antibodies to which you can attach toxins, you can attach drugs, chemotherapeutic drugs, or nanoparticles. So which of the directions, on just your opinion, is more interesting? Because immunotoxin, as we just saw, are immunogenic, but drugs or nanoparticles would not be.
Yeah. So we are also doing, I didn't show this slide, we are looking at very different approaches. So one is a naked antibody. The other approach, as you said, is an antibody-drug conjugate, where you attach highly potent drug. But to be honest, the problem with these patients, or even other cancers, is that these patients have developed resistance to chemotherapy drug. Even if you deliver a highly potent drug to the tumor, those cells, I think, are already resistant. And there has been a lot of work, maybe about 50 antibody-drug conjugates over the last 5-6 years that are being tested in the clinic. Only 2 of them are approved. One is using Herceptin, which by itself is a very good drug, and the other one is lymphoma. So I'm a little disappointed with the antibody-drug conjugate, but we don't know till we see it. But with the immunotoxins, they have a unique mechanism of action by inhibiting protein synthesis. So we believe that could be useful for patients who are resistant to chemotherapy drugs.
So mesothelioma, if I recall, is also associated with a lot of fibrosis that occurs in the area. So what happens to that when the patients are treated with immunotoxin?
So that's what we think. That's a classic teaching. But if you look at the PET scans and things like that of this patient, it's a highly metabolic tumor. But there is a residual underlying inflammatory cell populations of fibrosis. So when we look at some of these tumors in these patients, the responses are huge. But what is remaining could be an inflammatory component that we may not get rid of. And we see that in our mouse models where we take tumors, grow them in mice, where they just stay at a certain level. So it's possible there is some underlying fibrosis which remains.
So this is a tumor that can develop in mesothelial cells anywhere in the body?
Yes. So the most common is the lining of the lung and the abdomen. But I see a lot of patients who have mesothelioma of the lining of the heart, and also mesothelioma of the lining of the testes, because those cells have a mesothelial cell. But how asbestos gets there, I don't know.
Are those metastatic lesions, or is that asbestos?
No, the primary. They start out. I have a 16-year-old patient right now who just presented with thickness around the heart, nowhere else.
How limiting is the pleuritis that you've seen with the patients, you know, as a side effect?
How we manage it?
Well, how limiting has it been
Oh.
for the patients? I mean,
Yeah.
otherwise you have no toxicity with this?
So that is the main toxicity when we went up on the dose. So now we are at a dose which we feel can be given safely. Patients still do get pain, but it is managed easily. The other side effect that we sometimes see is that patients have increase in their weight, or they have swelling in their legs because of what we call as vascular leak syndrome. But that has not been limiting us in giving the drug, because it's pretty easily managed and self-limiting.
You know, I think particularly for the non-MDs in the audience, must gain some appreciation that I mean, this kind of clinical investigation being applied to patients with lethal toxins, you know, it requires, it's not just a matter, it's not a cowboy issue, you know, you go out and you just shoot. We can only imagine what it took to get proposals like this through an institutional review board, for example, let alone the enormous amount of work that has to be done to provide safety at every single step of the line. And I suspect that's where a lot of that 10 years went, correct?
That's true.
All right. Well, we'll have time for more questions of this later. Thank you very, very much.
That's good. Thank you.
Dr. Meltzer.
Yeah, sure. Okay, you got a pointer. Okay, perfect. Okay, so after that really elegant talk of patiently developing one treatment for one disease and improving it, I'm going to go the other direction into the very global world of cancer diagnosis. And it was a little challenging to figure out how to do that and still be demystifying, because cancer is such a complex, huge family of diseases with so many issues that it's hard to, it was hard for me to think of the right way to do this without obfuscating. So I'm going to try to give you the 50,000-foot view of the field that I work in, and try not to get bogged down into a lot of technical details, but rather the concepts that I think are driving the field forward, which is unbelievably rapidly moving. I mean, this is an incredibly quickly evolving field that has a huge amount of interest in it right now. So I think that anyone who, even the non-clinician, understands the concept of making the diagnosis of a disease. So if a patient has a problem, whatever kind of problem, in order to come up with the appropriate treatment for the problem, the appropriate advice for the patient, you need to know what it is. And if you imagine a patient with a cough and difficulty breathing, you might, do some X-rays and some bacteriologic tests and decide that they have pneumonia, and you'd want to know what kind of pneumonia they have and give them the appropriate treatment for that disease. So, that's where we'd like to be with cancer. Now, if you were to go back in history to probably, any time before the 18th century, you would find that cancer was really, thought of as a tumor, which is nothing more than a swelling. And people get swellings all over their body for lots of reasons. But I'm sure that astute clinicians, even in ancient times, knew that certain kinds of lumps in certain places with a certain feel when you touch them and certain symptoms in the patient were very likely to be a very bad kind of swelling that people died from. But it's remarkable how slow this was to develop into more precise ideas. So if you go back, initially cancer was thought of as an anatomic problem, where really what one would want to do is say, well, a tumor of the brain would be brain cancer, or a tumor in the lung would be lung cancer, and so on. So it was a cancer of an organ. And it really wasn't until, this is sort of absolutely amazing to me, that wasn't really until the late 19th century with the development of good microscopes and, importantly, the dyes that enable you to take sections of tissues and stain them and look at them under the microscope, that the simple anatomic diagnosis became supplemented with a cell-level diagnosis, a tissue-level diagnosis of histology. So instead of saying, yeah, this must be a brain cancer because it occurred in the brain, you also could look at the tumor, and the field of pathology has been devoted to understanding what these histologic patterns represent, and indeed the trained eye of the pathologist can recognize subtleties that others don't see that helps subtype tumors in the human body. So this is still the most essential basic mechanism for the definitive diagnosis of cancer. It's the basis for the recognition of all subtypes of cancer, and it's been a platform for continuous diagnostic refinement since these fundamental procedures were developed, 150 years ago or so. It's still the basis of all clinical decision-making. So when a patient is referred to the NIH Clinical Center for cancer treatment, essentially the first thing that is done is to try to get a hold of the paraffin block, which contains a piece of that cancer tissue, and we take it to the pathologist and confirm that they actually have a cancer and actually is the type of cancer that they think they have. So it's absolutely the basis of clinical diagnosis. It's the the basis of selection of treatment, and importantly, it's the basis of all clinical cancer research. So if you want to compare different drugs in 100 patients, you need to know that they actually have the same disease or more or less the same disease. So this is absolutely essential. And sometimes when I have a lot of pathologists in the audience, they think that some of the things that I'm saying is an attack on traditional pathology, and I in no way feel that way about it. This is absolutely the essential root of cancer diagnosis, combined with clinical and imaging studies and a few laboratory studies. But it's really the bedrock of cancer diagnosis. Now, I'll raise a question. I don't think I'm going to be able to answer it definitively, but one of the things that people are thinking about now, and I'll just sow the seed of this and try to come back to it later, is how's our progressive ability to subdivide cancers into finer and finer, precisely defined entities based on our traditional ways of classifying tumors, has it distracted us from the potential importance of commonalities among tumors which cut across different histotypes? You already heard that in the previous talk, that, well, mesothelin is a great target in mesothelioma, but it's also expressed in other tumors, and maybe what starts out as a mesothelioma treatment will turn out to be more broadly relevant. And so this is, I think, a little bit of a shift in our thinking. So new advances in cancer diagnosis are, we hope, based on an ever-deepening understanding of cancer biology and advancements in biotechnology, which let us see things in tumors that we were never able to see previously. And so the goals of this kind of work is to make a biologically informed cancer diagnosis, which will create the opportunity for biologically informed clinical decision making, and hopefully work towards precision cancer therapy. The term used to be personalized medicine, but it's been supplanted, I think, appropriately by precision medicine. I'm going to digress now for a moment and talk about the human genome, which is really the field that I come from. I'm a pediatric oncologist and a tumor geneticist, not a pathologist, and I think about things. I see the world through the lens of the human genome, more or less completely. And this has been an incredible process that went on from the discovery of the structure of DNA in the first report in 1953 by Watson and Crick, of course, to the report of the complete sequence of the human genome in 2001. And during that period, some amazing things happened. So there were tremendous advances in understanding genome function and molecular biology in a little less than 50 years that is shown here. But there was also tremendous technology development driven by all of this research, which has built momentum and is moving forward into clinical applications rather rapidly. So this has led to the idea that we can study the genome function with what people call integrated genomics. And basically, the genome is basically the sequence, the DNA sequence carried by an individual organism, in our case, the human genome. But it has many properties that we can measure. So we can look at, for example, the genes that are encoded in the genome and how they're expressed. We can look at the copy number that may vary from individual to individual. We can look at how DNA is modified and chromatin are modified by post-translational and biochemical processes. And we look at sequence variation between individuals, which can be things that you're born with or things that happen during one's lifetime. It's pretty clear that to understand how the genome works, we need to bring all of these different approaches, as well as others, into play. Now, I'm going to talk about, just very briefly, 2 slides on technology, because I don't think I can spend the time discussing any of these things in detail. But the 2 technologies that have dominated as we've developed the ability to analyze the genome, the first is the first to arrive at a truly high throughput platform was a microarrays. And basically, a microarray is nothing but a solid support on which, in the modern era, millions of synthetic oligonucleotides are attached. And these create probes which can be used to analyze almost any feature of the genome. And this slide just shows the evolution of these from when we started working with this technology in '96, and we had only about 2,000 spots, until the present, where we have, in the span of about 10 years, we went into tens of millions of probes. And it's a very powerful and still useful technology, rather routine at this point. The other thing that's happened is this incredible change in the ability to sequence genomes. And this slide shows how this happened. And unquestionably, this was driven by the Human Genome Project and by the recognition throughout the whole world of biology, because we're talking DNA, it's something that applies to all fields of biology, microbial, plant, animal, you name it. So there's been a huge motivation to get better at sequencing genomes. And this is, in fact, what's happening. This shows the cost per genome compared, by the way, to Moore's law, which is the ever-falling cost of computing power, which used to be. So DNA sequencing passed Moore's law abruptly here about this point. And this was with the development of new sequencing technologies. And the goal has always been the $1,000 genome. And as you can see on this graph, it didn't we're into the several $1,000 genome by 2014, but we actually are looking at fall towards the $1,000 genome in the next year or two. There's a catch. You have to have a huge sequencing center which can spend huge amounts of money sequencing many thousands of genomes to get that degree of savings. But I have no doubt that disruptive technologies will continue to be developed and that genome sequencing will be as routine as a blood count at some point in the future. I can't say when that will happen, but I think it can be predicted with some certainty that it will happen. Now, back to how these things relate to cancer and back to cancer as a genomic disease. So that's, of course, what's excited me since I was old enough to understand this concept, which turns out not to be that new. And this is one of these very humbling quotes from the history of science from Boveri, a German scientist who in 1992 said that a statement which was incredibly prescient. So in every cell, he said, there's a specific arrangement for inhibiting and definite chromosomes which inhibit division. Tumors would arise if those inhibiting chromosomes were eliminated. So this is the prediction, the correct prediction of the existence of a tumor suppressor gene. But what's fascinating is that although this idea came out at the turn of the last century, it wasn't accepted by the scientific community readily for quite a long time. And I have a contrasting quote here, which I love, from the Nobel Prize lecture of the tumor virologist Peyton Rouse, who makes a conflicting statement. So he goes, what can be the nature of the generality of neoplastic changes for the step-like alterations that they frequently undergo? A favorite explanation has been that oncogenes cause alterations in the genes of the body, somatic mutations, as these are termed. But numerous facts, when taken together, decisively exclude this supposition. Well, there you have it, 64 years after Boveri, strong opposition by powerful people in the scientific community who were wrong. So that's an interesting juxtaposition. But I can tell you that since 1966, using simpler and gradually more advanced technologies, that Boveri was proven right over and over again. And we now have a huge amount of data supporting this concept that cancer is basically a disease of the genome. Many aspects to it that may not be easy to capture from the genome, but it's certainly a disease of the genome. So the way I would put it is I feel comfortable now taking it as axiomatic that alterations in the cancer genome substantially determine the malignant phenotype, and that characterizing these will hopefully yield correspondingly substantial insights into tumor biology with clinical implications, one would hope. And so just to give you two pictures, which I think translate this into something that anybody can relate to. So here are we think of genome sequencing as being a modern technology, but in fact, the oldest whole genome scan was the chromosomal karyotype. So here you see the normal 46 human chromosomes, the 22 pairs of autosomes, and the sex chromosomes looking all lined up and neat in a metaphase cell from a normal tissue. But what happens if we look at a typical adult carcinoma? It might be something like this, and where we see all sorts of really terrible things. We see chromosomes that obviously are structurally abnormal, some that even are forming rings, some that even a trained cytogeneticist can't tell by looking at them what they are, some that are missing because they're scrambled up into all of these other chromosomes. And so you can say that this genome is really screwed up, it's abnormal, it's got a lot of changes. So the challenge has been, out of all of this, to figure out what's important in the cancer, because there's some stochasticity to these processes, there's no doubt about that. So then we come to the field of cancer genomics. So instead of looking at the normal genome, we look at these same variables with technologies that are suitable for studying the cancer genome. And I don't have enough time to walk through all of these, but I'm going to take a couple of examples. Gene expression, DNA methylation, and sequence variation is something that I can talk about today. And although I'm going to keep this very general, I'm going to use some examples from very specific diseases that I've worked on because I think they're fun to try to understand. So the first question that we faced in the early genome era was, can cancer types be defined by their gene expression profile? Well, this seems terribly obvious now, but it really wasn't at that point. So are all the genes expressed in the same way in every cancer, or are they different? And we started working on this using array technology in the 90s. And I'll show you a couple of slides from a study that we did in 2001, in which was one of the first to classify similar looking tumors. So these 4 types of cancers all look about the same under the microscope, and they're frequently confused. And Javed Khan, who was a fellow in my lab at that time, decided to set about separating these what are technically called small round blue cell tumors using microarrays. And this is just shows the result of that kind of analysis. The separation here between different tumor types shows their pattern of gene expression. That's 4 different tumor types. Kristen Baird in the lab later looked at 11 different tumor types. And here what she's done is for all of the tumors labeled at the top here, have identified the genes which most characteristically define each of those tumor types. And what was sort of exciting and maybe a little misleading at the time is that when we looked at one of the tumors, and I'm going to focus on that for a few slides here, it's called gastrointestinal stromal tumor. When we looked at just what's in that list under the column that's headed just here, if we look at the genes that are in that list, we see that the top one is the KIT oncogene, which turns out to be both a diagnostic marker and ultimately a therapeutic target for most GISTs that occur in adults, right at the top of our weighted gene list of the genes that most specifically characterize it. And in fact, all sorts of good biomarkers have come out of these lists. And one would hope, looking at this, that maybe some therapeutic targets as well. That's turned out to be challenging because it isn't easy to treat specific genes just when you want. Think of them as a possible target. So there are a lot of implications from this type of work, which is many thousands of publications in the literature now. Probably most importantly, it's a powerful tool for class discovery, which is still leading to the recognition of molecularly defined subclasses of tumors. The data can be used to develop diagnostic and prognostic clinical tests. And you can identify individual genes which can be readily transitioned to clinical assays such as immunohistochemistry, flow cytometry, or RT-PCR. So I'm going to talk a little bit about gastrointestinal stromal tumor and how we've been able to further fractionate these some unusual subsets with integrated genomics. So for those who haven't heard of this tumor, they're derived from a specialized cell type called the interstitial cell of Cajal, which is a pacemaker cell in the lining of the gut. So this is what causes the synchronicity of peristaltic contractions in the gastrointestinal tract. And it's been a model for targeted therapy because, as I said, most of the adult cancers have activating mutations in a tyrosine kinase called KIT. And if they don't, they tend to have one in another kinase called PDGFRA. And quite impressive clinical results are found in some patients. But what we got interested in was the fact that not all GISTs carry these mutations. Particularly, we got interested in what we call the pediatric and wild-type GIST clinic wild-type because they didn't have those kinase mutations. So there's a subset of patients that lack the KIT and PDGFR mutations. They're often pediatric and hereditary. And so Lee Hellman helped set up with a number of colleagues and collaborators an interdisciplinary clinic here, which brought patients to NIH to study this rare disease. Interestingly, they're mainly gastric tumors. Hereditary cases were recognized pretty quickly that they could be associated with other tumors, particularly paragangliomas, which are tumors of the peripheral nervous system, and they may be caused by mutations in genes which encode an enzyme in energy metabolism that occurs in the mitochondrion called succinate dehydrogenase. In some cases fit a very rare syndrome called the Carney triad, where patients get both have a predisposition to paraganglioma, and to rare tumors called pulmonary chondromas. And so we set up this clinic here at NIH to try to study these patients specifically. And this just shows a slide from Markku Mietinen, a terrific pathologist. And this was a clinic has been very fun to participate in because we had such a great team of experts here at NIH. And Markku is an expert pathologist. And what this slide really shows is the disappearance of SDHB staining in a tumor that's SDH deficient. And this is actually the simplest diagnostic test to recognize most of these patients. Now, let me introduce the concept of epigenetics. So genetics is information that's transmitted from parent to child through the DNA code. Epigenetics is something that is the property of particular cells based on their lineage and development. And this plot from Waddington, who was a very clever guy, and I love the illustration because it's easiest to understand. So what this is, so if you imagine this is a cell here occurring in the state of a fertilized egg. It's pluripotent. It could develop into any tissue or organ in the body. But as development proceeds, it finds its way towards these specific valleys, which may continue to fork and branch into the different cell types that are found in the human genome. And these have different epigenetic states based on modifications of DNA and chromatin and modifications ultimately in gene expression. So we decided to look at that angle, the DNA methylation angle, as well as the DNA sequence angle, and put these things together with the gene expression in our clinic patients. And so I'll show you what we see here. If you look at the left panel first, each of these little spots is basically represents the epigenetic state as measured by DNA methylation. And you'll see two clumps of spots, these down here at the bottom and these up at the top. And these are all just tumors. And what we found, these are all the tumors that have mutations in one of the 4 SDH subunit genes. And these are all tumors that have mutations in one of the signal transduction pathway genes, which would be KIT, PDGFRA, BRAF, NF1, the neurofibromatosis gene. They're really completely different. And this was kind of a shock. I honestly was not expecting that we'd see something. And this is probably the most clean binary separation within a closely related disease group that I've ever encountered so far. These are tremendously different. And in fact, immediately we can diagnose at the molecular level a patient with SDH deficiency based on this phenomenon. And this is just a 2-dimensional heat map, which shows the same thing and shows that primarily what we're seeing is hypermethylation. So red means more DNA methylation. Each row represents a different point in the genome, although there's a little bit of loss of methylation in some places. It's mainly a hypermethylation phenomenon. Now, this is showing these 2 subtypes of tumors, which we're now trying to diagnose. I'll show here what that group of tumors with hypermethylation look like compared to normal tissues. And the point I want to make here is that these different colored blobs represent different normal tissues and that they cluster rather closely together. And no tumor ever looks exactly like a normal tissue when you look at its epigenome. They're always different. And they resemble no normal cell in the body. This, to me, is an absolutely fascinating thing. So Keith Killian, a terrific staff clinician in the lab, did this work. But we were left a little bit puzzled. As we were doing this, we we're developing next-generation sequencing technology so that we could sequence all of these genes at one go on one of the new kinds of sequencers. And we had figured out what almost all of them, the mutation was in almost every one of these, but we were left with a pretty good-sized fraction of this group for which there were about a third, not quite a third of the cases for which we could find no mutation for any of the genes that we knew were connected to this pathway. So we did something different, and we decided, could we split this group of patients even further? And and I'll show you how we did that in a moment. And in terms of the mechanism of this, I just want to say that we don't really know for sure, because it's very hard to study these tumors, for which we have no good model systems in cell lines or animals. But this just shows here on the left the SDH tetramer embedded in the inner mitochondrial membrane. And its main job biochemically is to convert succinate to fumarate in the Krebs cycle. And it turns out that there's an enzyme, and there are actually several enzymes related to epigenetics, which produce succinate as a byproduct. And one of them is an enzyme called TET2, which is interesting because it's known to actually be a true tumor suppressor gene in leukemias. And what we think happens is that succinate builds up when you don't have a functioning SDH complex in the cell, and that it poisons TET2 and blocks this reaction, which is the conversion of 5-methylcytosine to 5-hydroxymethylcytosine for maintenance demethylation. So we think methyl marks just build up in the cell from this biochemical perturbation. So our conclusion at this point was that these, which I'm calling SDHX, because they can have a mutation in any of the 4 subunits, so that these SDHX mutant tumors have the deleterious mutation in one of these genes. They have frequent loss of heterozygosity of the second allele, so they have no functional SDH complex at all. And which, by the way, it's an amazing thing. They think that the cell doesn't just die, but actually is activated to develop into a tumor. They have this pervasive remodeling of the epigenome that I showed you, and they have relatively few copy number changes. They're pretty simple compared to other cancers. So, as I said, we found that most of the SDH deficient tumors were readily classifiable by DNA sequencing, but there was this group with no molecular diagnosis, and we're trying to figure out what's going on in these patients. And we resequenced them, and we looked manually at every bit of data to try to figure it out. And we actually did manage to find subtle mutations in a couple of cases, but most of them we couldn't find anything. So we decided to look at the global level again. And we did, in this case, a differential methylation analysis on the DNA and gene expression data to the extent we had enough samples of these SDHX wild types, so ones where we could find no mutation, to those which had mutations. And we got an amazing result. Again, totally unexpected. And really, the reason we could find it is we had the right patients to look at. We did this, and I'll show it on a plot that we actually sort of made up for this study, which we call a Janus plot, because it looks 2 ways. It looks like at methylation and expression of genes at the same time. So here's a paper that we just published, and what we found is looking across the whole genome, there was one point in the genome that showed increase in methylation, which is thought to be, in many cases, a silencing mark. It slows down gene expression. And in this case, we see at the same point in the genome, in fact, a loss of gene expression. So we have exactly the signature that you would find in a very focal DNA methylation, which silences a gene. Well, would anybody care to guess what kind of gene it is? This was, I guess, one of those things that you might have guessed ahead of time. It turns out to be right on top of the SDHC gene. So when you expand out from this point, you see that this region of locally increased methylation is right on top of the SDHC gene at the promoter end of the gene and spreading a little bit into the gene. And when we looked carefully at the gene expression data, we found indeed convincing evidence, and that's just shown here. These are different probes on a commercial microarray that show loss of SDHC expression in a series of samples that show this hypermethylation. And what was fascinating was that we found that this was essentially in every patient in whom we could not find DNA level mutation. And so we term this an epimutation, and so we were able now to make a definitive diagnosis of what was going on at the molecular level in now almost all of the patients that we've seen in this clinic. Different diagnoses that may be clinically rather different in how they evolve, but in these very closely related tumors. So this is splitting to the nth degree. But we found that in the tumors which lack a mutation in an SDH subunit, SDHC is typically inactivated by what we term an epimutation. So we don't understand the biochemistry of this and why it happens in detail, but it's clearly pushing these cells down the same final common pathway of pathogenesis, which is loss of SDH function. So, and then finally, we did also realize that these patients with SDH mutation are also the ones who are susceptible to these other related cancers, paraganglioma and pulmonary chondroma, which is one of the Carney triad. So we think that this is actually the cause of the Carney triad, most likely, although it'll require some time to validate. So I'm going to switch gears now and talk about protein alteration mutations in cancer in general and the problem of cancer therapy. And we have a ton of data. Everyone's probably heard of the TCGA project. Literally thousands upon thousands of tumors through that project and other international projects have been sequenced. There's a huge amount of data available at this point. And so things we thought were impossible are now routine, and there are massive quantities of data. One of the things we've learned is that tumors vary in terms of the number of protein-altering mutations, from tumors that are exposed to carcinogens, such as UV exposure or cigarette smoke or the like, may have really thousands of mutations, whereas many pediatric tumors, as a rule, contain less than 20 mutations. And there are some cancers that appear to have only 1 or 2 mutations, and some puzzling ones where we can't find anything yet. And I just gave you an example of one where we couldn't find anything by sequencing of the DNA alone. So there's a huge variety in complexity. But I'm just going to tell you a story of one simple example, which many of you may have already heard about, and that's chronic myelogenous leukemia. So going back to looking at the chromosomes of cells, in 1960, Knoll and Hungerford found that these tumors and this is a chronic leukemia that evolves rather slowly, they all carried an unusual chromosome by just staining the chromosomes. They called it the Philadelphia chromosome because they worked in Philadelphia, but they couldn't, this was before banding was invented or perfected, and they couldn't tell really what it was. And then Janet Rowley at the University of Chicago in '73 figured out it was a translocation, that is a fusion, which joined 2 chromosomes, chromosome 9 and 21. And then Gerard Grossfeld in mid-'80s, he showed that this was a fusion gene, that the 2 breakpoints created a chimeric gene, something that never occurs in any normal cell. It's a cancer-specific gene that fused 2 genes, one called BCR and the one called ABL. And ABL, it turns out, is a tyrosine kinase. Well, this information was really exciting because it gave us a specific molecular pathogenesis for a relatively rare but important leukemia. And then that information, basically sat around for a while until 1996, when Brian Drucker, who really made this his cause, said, well, look, if it's caused by, the disease is caused by a kinase, there should be a drug that blocks that kinase. And he worked long and hard to come up with collections of drugs, and he found one called imatinib that actually is effective in inhibiting the BCR-ABL fusion oncoprotein. And the result of that is that the transformation of the treatment of CML forever. And I'm not going to go into the clinical results except to say that they've been extraordinarily good. And now there's all sorts of other related drugs to deal with issues that arise in treatment in unusual patients, problems of resistance, and so on. But it's completely transformed this field. So the reason I go through this in some detail is to say, well, does CML define a paradigm for cancer cure? And a lot of people thought that, and many of us really desperately hoped that that would be the case. So what is that paradigm, and how would we know when we're encountering it? And are there other examples? So you want a specific driver gene that's needed for the cancer cell survival that drives the growth of the tumor. And you need a therapeutic agent which can target that gene. Simple. So to put it in a kind of graphical format, the idea is that we have some specific tumor biology that determines the outcome, a leukemia, but we're going to make a therapeutic intervention that's going to prevent that biology from expressing itself. Now, of course, even then in the mid-90s, when these things were starting, we knew that all cancers wouldn't have the same driver mutation, and that we'd have to come up with different kinds of biology and biology-specific interventions. And so BCR-ABL might be one, or an EGFR mutation, epidermal growth factor receptor, might be another. And that was the simple idea. Well, have we been successful in doing this? Yes, I can definitely point to some successes, and some of the more important ones are listed here. There are many more in various specific situations. So there are other things that are close to the CML-like situation. But so this comes with buts and ands. And so the but is that in most common adult cancers, CML-like results are not achievable. We now know that with a single drug, even when a druggable target gene is present. So there's something special about this leukemia case that makes it a little bit easier to treat. And a large proportion of human cancers, and this is the more depressing aspect actually, a large proportion of cancers do not have a gene which has been activated by mutation which fits the CML paradigm. And that's a tougher problem to think about how you're getting around. So, in addition to not getting terrific responses with single agents, there have been lots of instances of resistance developing by many different mechanisms, mutations in the target gene, activations of alternative pathways, you name it. And this is now a relatively sad and sobering saga. But there is an and to this story. So first of all, many of the altered genes that have been successfully targeted in cancers where they occur frequently are also observed at low frequency in diverse other cancers and or rare cancers. And so this gets back to the point I made at the very beginning. So could known targetable mutations be useful as molecular markers to guide precision therapy out of the disease context in which they were originally validated? The biggest example, the most frequent example of this is the BRAF gene, which is a kinase that's activated very frequently in melanoma. But it turns out that the exact same activating mutation, which is a V600E in BRAF, occurs in lots of different kinds of cancer. And in some of these, there's reason to think that BRAF-targeted therapies may also be effective. And so this is kind of where we're going. What cuts across lots of different tumors and goes beyond the histologic view of cancer. Okay, so I have one parting shot here. So as we've been sequencing more and more tumors, it's now become practical to sequence DNA from small clinical samples quickly and accurately. And you can do it on any scale, whether from the grand scale of the whole genome to individual genes. sequencing of a panel of so-called driver genes which could be targeted for therapy or which would aid definitive diagnosis. It's now clinically accessible and kind of routine. The broad utility of tumor DNA sequencing is currently a subject of ongoing clinical research, but sequencing is already standard of care in specific clinical situations. Now I want to make a quick comparison of this model of tumor progression, which is a sort of an old model that you start off with a normal cell, which then gets successive mutations in genes until you finally get invasive cancer. And we know now that this linear way of thinking about cancer evolution is not really correct, and that a better way to think of it is in terms of branching, polyclonal disease. So if that's the normal cell, then tumors will pick up changes and bifurcate into clones, some of which may die out. And here in blue is one that becomes dominant in the tumor when it's diagnosed as a tumor it grows. And there are some other secondary clones which are actually not that rare. Now, if you treat the patient, you may find that the tumor recurs and that a clone is different when it comes out. And this concept, which is not that new, I think is turning out to be actually really important. Why? Because the clonal heterogeneity of tumors can be quantitated really well by tumor genome sequencing. This has been best achieved with leukemias, but I'm willing to bet that it will happen in other cancers as well. And this is my last slide, and I'll conclude with this. This is my vision of the future of the molecular characterization of tumors, which is that at the time of tumor biopsy, when a patient presents to the oncologist, in addition to the usual things that I showed you that we were doing 100 or more years ago, and a few immunostains, we'll also be gathering tumor nucleic acids for tumor normal genome sequencing, RNA sequencing, methyl sequencing, and from that we'll get all of this information, accurate classification. We'll find outlier gene expression, which may be good for targeting in one way or another. We'll find out about gene amplifications and small mutations. We'll learn about neoantigens, which is a really cool topic now for antigens that never exist in nature but can occur in a mutant tumor cell. And all the other things I've talked about, translocations, we'll try to be able to predict pathways and carry out this clonality analysis, which will enable us now to think of not of a cancer as something you've got the day it's diagnosed, but as a process that's going to change as the patient often lives longer and longer with the tumor. So at recurrence, these aspects of this diagnostic process will be repeated, and the tumor will be reevaluated and retargeted based on the active proliferating clones at that time. At least this is a little bit of a vision, and we don't have most of the information you need to do this, and it's still too costly to do, but I think that there's little doubt in my mind that this is going to happen, and it's already happening in the leukemia world, and I think we'll see this expanding more and more. So that's what I had to present, and I'd be glad to take questions. Thanks.
Thank you very much, Paul, for an insight into the brave new world.
When you work with cells in vitro, it's clear you can sequence because they're all identical, clonal. But now you're taking a tumor which has all stromal cells infiltrating lymphocytes, endothelial cells, plus you have tumor cells which could be highly heterogeneous.
Right.
My question is, how are you sorting out which signal is coming from which type of cells, and how are you establishing signature for that?
Right. So, I mean...
I have a second question.
Okay, just really quickly. I mean, you asked a question that opens a big world of other questions. But what I can say is certain things you can tease apart. So, in fact, we do this kind of routinely for aspects of the signature of normal cells that are mixed in. That's one thing for at least at the gene expression level. But at the mutation level, we try to estimate the percentage of tumor cells, and you can just overcome that heterogeneity by sequencing more, because the normal cells generally will not carry a big mutation burden. The mutation burden isn't zero, but it's easy to spot. So there are some strategies. A bigger problem, I think, is then the local heterogeneity, sort of microscopic stuff, is the heterogeneity geographically in a tumor that's large. There's no reason to assume that a tumor here is identical to the tumor that's a few centimeters away. That's a big challenge, and we know that's true already for some tumors. That's just a relatively new field.
So my second question is to both speakers. Is there any genetic signature of mesotheliomas? Because the first speaker just mentioned BAP1 protein involved, but what about other changes? What is known?
Well, I don't know. I think the fair thing is to say we're working on this, and we don't have all the answers yet. But there are some genes that are known to be mutated in mesothelioma. But I think we're at an early stage.
Yeah. And especially given our mesothelioma clinic, Paul and I are very interested in looking at young patients with mesothelioma, less than 20. We frequently see young kids with mesothelioma, so I think that might hold important.
Yeah,
So
might be important.
P53 is very uncommon in mesotheliomas, but we see P16 that is present in about 80%. And then we talked about the BAP1. Not really. NF2 in some of those.
Regarding the SS1P treatment, what is the immune reconstitution like for these patients, and do they usually receive transplant afterwards?
So the patients get lymphopenia, but they do not get a decrease in their neutrophil. And the lymphocytes usually recover within it's variable how much treatment they get, but within 3 to 6 months, their lymphocyte counts are back to normal. There's no transplant. It's just a lymphodepletion, but the lymphocytes recover on their own.
Okay.
So the recent studies by Dr. Vogelstein from Johns Hopkins makes a unique observation that at the advanced stages, the cancer formation is by bad luck. So at what stage we start from a predicted phase versus this uncertain domain?
Right. I think that, in fairness, and in fact, there already have been some commentaries on this, that paper, which is a very nice paper for giving the overview of the disease, he didn't really say that the bad luck. That was the press translating that paper into, something for the public consumption, because it gives the misimpression, because you make your luck to some extent with things like what cancer risk factors you expose yourself to. Most notorious is smoking, but lots of other things. So there is clearly a stochastic element to this, but it's one that can be affected by genes. It can be affected by lifestyle. So you have to there's luck and there's luck, right? So that's a kind of a complicated topic to simplify into, well, it's just bad luck. You may be a smoker because you have a gene that predisposes you to smoke. That's bad luck.
Sorry. I just wanted to tack on a question to her question earlier. Have you had any patients that were already immunocompromised, HIV positive patients, for example?
In general, so the question was, did we have any patients who were immunocompromised, such as because of HIV? But in general, for these clinical trials, we exclude patients because we're worried that if we further lymphodeplete them, that could make the disease.
So we heard
wondering, you did not have any opportunistic infections during.
Been hearing a great deal in the past, I don't know, 5 years or more, about the Cancer Genome Project, in which a large number of cancers are being sequenced. Can you tell us what the general status of that project is, and what have we learned from it?
Wow. Let's see. Well, the general status is that funding for that project is being concluded, and the projects are being published. And there's already a huge number of papers that have come out. And most of the major diseases have publications. One of the other nice things is that the data is available publicly. The process data and the raw data is also available. There's a few more hoops that you have to go through. So this is the kind of data which you really can't squeeze everything out of it in that first overview paper. So there's a lot left to discover. But what I would say is that there are a number of instances where diseases that were considered to be one have now been clearly split into definitive subtypes. This is particularly true in some of the brain tumors, where a really good example is medulloblastoma, which now can be really broken into several subgroups based on its gene expression profile, which is related to its mutational profile. So there have been many, many such instances. I would say also the other thing that comes out is that when you find a gene that's unknown to be mutated in cancer, and you find it at 15 or 20% of some cancer, then you take it much more seriously when it's mutated at 1% or 0.5% in another cancer. And so it's tending to build these bridges of thought across the whole field, which I think will take a long time for us to sort out, because we don't know how this will add up in terms of potential therapeutic options, or the opposite, knowing that a therapy won't work. And, sometimes the presence of a mutation, a good example being
that's an ongoing project.
Right. So that's ongoing.
All right. Any more questions? If not, thank you both very much for a very exciting afternoon.